welcome to my channel Srinivasa Biology Tutorial. For this section to discuss NCERT class 12th, the chapter Sexual Reproduction in Flowering Plants, part 2, that is in a microsporogenesis. Let us, here it is microsporogenesis. Here, as the anther develops, the cell of the in sporogenous tissue undergo in a meiotic division form as in a microspore tetrads. For example, here when you take as in one particular in a that microspore, that anther after the microsporogenous tissue develops to form as in therefore tetrads. Okay, in this what would be the ploidy of the in tetrads, which has in what in completely their n condition. They having as what in a n condition. Next, after as each cell of the sporogenous tissue is capable to give rise to as in a microspore tetrads. Each one the potential pollen grain or microspore mother cell. So each one to get form as what in this in a 4 tetrads. Here in this each tetrad it is become as in what developed as in a here pollen mother cell. It is to develop as what in a the pollen mother cell. See that in the process of formation of the microspores, the pollen mother cell PMC, that is in what PMC, through that in the process of meiosis is called as what this in a microsporogenesis. Here microspore genesis means to give as a birth. Here this microspore to give as in a birth for in this their spores, that is called as what in a microsporogenesis. Let us the microspore as they formed or the arranged in the cluster of 4 cells, that is in a microspore tetrads. When you observe as in that here one figure, it is appear as in a complete the 4 tetrads. Okay, in the anther the mature the dihydrate the microspores that dissociated from the each of other developed as in a pollen grains. After in this each one to get after to develop as what it is become as in a completely in pollen grain. After to get develops as in a pollen grain. Inside of each microsporangium, the several thousands of microspores or pollen grains are formed there in a released within the dehiscence of the anther. So once here when you take as in one particular anther, after the inside here the number of pollen grains are to get mature. Here what happened, then after these are completely to dehiscence, then to release as in the outside of the anther. Then what are the in a pollen grain? Now see that in a pollen grain, the pollen grain represent as in a male gametophytes. If you touch the in a the open anther, hibiscus, any other flowers, you will be find the deposition of yellowish powdery pollen grains. You the fingers, the sprinkle these pollen grains or drop of water taken the glass slide and to observe under electron microscope. When you observe as in that in microscope the microspore, you will be the really the amazing. So when you take as in that pollen grain and be kept on the few add within a few drops of water under after you observe as an electron microscope, it is appear as in a there clear visible is will be there appear how the pollen grains will be get germinates. Will be the really the amazing the very they completely architecture, size, shape and colors and designs you see the pollen grain from different species. When you observe as in the different species, they appear as in a different colors and shapes. And let us see that in a one particular pollen grain. Here they given as a clear the pollen grain. In this pollen grain around as in 25 to 50 diameters. Let us see here the pollen grain are generally the spherical shape and measuring about as in 25 to 50 micrometers diameter. So when you observe as in one particular the pollen grain, it is it completely it is 25 to 50 diameters, micrometers in diameters. It is in a permanent the 2 layers wall. It contains as a 2 layers as present. One is in a outer layer is called as an exine and it is made up of as a sporopollen, which is in a one of the most resistant organic material known it. So when you observe as in it is the exine layer, it is what? It is in a exine layer. It is there completely they are made up of as a sporopollen. When the sporopollen is present, then the pollen grain to get easily survive as in the extreme conditions also, means high temperature regions also they can easily they get survive. Next, It can be withstanding the temperatures and the strong acids and alkalines. Just before I say that one only, here the withstands as in high temperatures, as in strong acids and alkali. Here no enzymes to get degrades the sporopollenin is in a far known. So the no enzymes also that not degrade about as in a that enter the in pollen grain. Next, the pollen grains or enzymes has in a the prominent the apertures is called as in a germpores. When you observe as in that particular the pollen grain here they having as in the small pores as present. These pores are called as what? In a germpores. For example, see here I draw here that pollen grain. In this pollen grain, here the small pores are noticed. Here these pores are called as what? These are in a germpores. These are completely germpores. Okay, and after next germpores. When the sporopollenin is the absent, so when this enter germpore region here, what happened? The sporopollenin is absent. The pollen grain is well the preserved and a fossil because there presence of the sporopollenin. So the number of fossils they get completely the preserved in many years. Why? Because to development of the number of species. Okay, how do they are complete the pollen grains will be they are preserving here because the sporopollenin is there protecting. The exine exhibits as in they are facilitating the area of the patterns and the designs. It is like it shows as in a particular their designs also. Why do you think that an exine should be there hard? What is that in a function the germpore? So here the hard code to get is to protect the in a pollen grain, and they are asking about as what is the function of germpore. The germpore function is what to get a completely to exchange of the gases or to exchange of the nutrients, whatever this completely get helps. Next, the inner wall layer of the pollen grain is called as what? That is the intine. So the inner wall layer also is present here. See here, inner layer also they are present. This inner layer is called as what? It is in a intine. This inner layer is called as what? That is intine. And this intine layer here, this is in a thin and continuous layer made up of as a cellulose, so that and pectin. So this layer is made up of what? It is made up of as in a the cellulose and then pectin, cellulose and pectin. And then the cytoplasm of the pollen grains is surrounded by as in a plasma membrane. So this entire the surrounded by as in what? The plasma membrane is present. Now, when the pollen grains is a mature that contain as a two-celled condition, one is a vegetative cell and the one is in a generative cell. When you take as an one particular the mature pollen grain, it has show as a two layers. Sorry, it show as a two cells. One is in a it is in a generative cell and it is completely what? Vegetative cell. Two cell condition. One is in a here generative cell and it is in a vegetative cell is present. Okay, next. After the vegetative cell, here vegetative cell it is in a bigger. Mostly the vegetative cell is bigger and abundant for food reserve and a large irregularly shaped nucleus. So here in this vegetative cell, when observed as in this vegetative cell, see it is in a the large shaped and irregular shape of the nucleus also they are present, and here completely reserved as what? Food materials also. It is completely to reserved as any food materials. Next, here the generative cell is small and the floats of the cytoplasm of the in a vegetative cell. So it is in a generative cell. It is what? Generative cell. It is in a small size. They are completely freely floating on the cytoplasm. And the spindle shape of dense cytoplasm and the nucleus is present on here. Yes, dense cytoplasm as well as in a, the nucleus also they are present. And over 60% of the angiosperm, the pollen grains, they are shaded as in a two-celled stage. So when you observe as in a maximum the 60% of in a angiosperms has in a the pollen grain has in which condition? It is in a two-celled condition. It has in a two-celled condition. And in the remaining species, the generative cells will be divided as in a mitotic division to give as in a two male gametes before the pollen grains. It has in a three-celled condition. So after it is to divide as what? It is divided as in a to get form as in a to mitotic division to give as in a there are two male gametes. So it is a one male gamete, it is a one male gamete. So first male gamete is in a second male gamete. So completely get as how many cells? That is in completely three cell condition. That is in a three cell conditions. And then the pollen grains are mostly get using as in a They are different ways, and their causes and several their diseases also. Let us see that one here. Mostly here, the pollen grains, many species they cause several allergies and bronchial affections, and some peoples often leading the in a chronic respiratory disorders like as an asthma and bronchitis, etc. So the number of pollen grains they cause it as a number of diseases like as in respiratory diseases and chronic disorders, asthma, bronchitis, like okay, and it may be mentioned in a Parthenium or nothing but as in a carrot grass. Parthenium histophorus carrot grass. It came from India into yes, carrot grass came into India as a contaminated in a wheat seeds. It is become their unambiguous occurrence and causes as a pollen allergy. So the carrot grass they come from Australia with the cultivated of any wheat seeds. Okay, this is next. After in this pollen grains has a rich nutrients. Here the pollen grains has a rich nutrients. It has become as a fashion the recent years get used as any pollen tablets and then food supplements. The most of the pollen grains are using as in the pollen tablets and food supplement purpose also we are using. And in some western countries as in the large number of pollen grain, the pollen products from the tablets and making the syrups available for the market. So in that some in a western countries also the pollen grains get using as a making the syrups and tablets. Okay, and the pollen composition here conception has been that climbed to increase the performance of athletes and race horses. So here the number of athletes also these pollen grains were taking as energy drinks for their in a their race horse. Okay, next after when once that sheet the pollen grain has in the land the stigma of the before they lost the viability they have any bring about as any fertilization. So once they are to release as any the pollen grain to reach as any stigma they lost their viability because about that in a fertilization. How long you think as any pollen grains they retain the viability? So if it is the long their viability is there, how do they survive? Here the pollen when the period of which of the pollen grains the remain the viable heavy the variable and the some extents depends on the any the prevailing temperature and humidity. So in this when they are under the pollen the sporopollen is present, the sporopollen they are completely to get protected as in a pollen grain. And in some cereals such as in a rice and wheat and pollen grains lost the viability around as in 30 minutes they are the release. This is a very important. Here the pollen viability is 30 minutes only in that wheat and rice. And in some members of like as in a rosaceae and leguminosae and solanaceae family, in this completely families that in a pollen viability they having in completely in a months, few months. Okay, the pollen viability is around as in a months also. And you have in either hard in a strong this completely the semen sperms many mammals including as any humans for as any the artificial the insemination. Yeah, in that sperms also we are artificially with their insemination will be there going on here. And it is possible to store the in pollen grains in large number of the species for years in liquid nitrogen in minus 190 degrees centigrades. Okay, here most of the pollen grains stored as in a the different chemical compositions we are using, that is in a liquid nitrogen minus 190 degrees. Such stored pollen grains are used as in a pollen banks and similar to as in a seed banks in the crop breeding programs. So here there's a number of pollen grains are complete they are storing for as in a that pollen grains in where the complete the pollen banks or in a seed banks, which purpose it is help for as in a their crop programming purposes. Okay, this is in a completely in a that male the androecium and to get formation of in a pollen grains. Okay, let us to discuss as in a remaining to discuss in next class. Thank you for watching.